So let's review some key concepts that we have been discussing. Remember that our first line of defense against cancer is the transformed cell itself. It's kind of a weird way to say it, but we rely upon the trillions and trillions of cells in our body to be able to manage themselves, right? That means if they encounter problems that they cannot fix with their genomes, such as mutations, then they should do the right thing and undergo apoptosis. And so that signal for apoptosis is coming from within the cell itself, so an intrinsic kind of signal, and it's mediated by proteins such as p53, the guardian of the genome. So our second line of defense then, if you have transformed cells that unfortunately have gained mutations as part of their transformation that are somehow interfering with them undergoing apoptosis intrinsically, right? They're no longer managing themselves. Then our second line of defense, if tumor suppressor gene pathways like p53 are lost, is our immune system then. Our immune system has to be able to detect the cancer cell and then encourage the cancer cell to undergo apoptosis. So this message for apoptosis is coming from the outside of the cell. So it's an extrinsic signal. So this involves actions of immune cells like natural killer cells and T cells interacting with a cancer cell and sending appropriate molecular cytotoxic messages that trigger things like caspase pathways to kick off inside that cell. So we've been talking about the immune system and saying that in order then for cancer to succeed in getting around the immune system, essentially cancer cells are stumbling upon mutations that allow them to hide from the immune system. Some of the things that we talked about have to do with understanding some basic concepts of how our immune system polices the cells in our body. It's up to our body cells to constantly be presenting on their exteriors some evidence of what's happening inside of them. Oftentimes, this is in the form of presenting small little peptides of recycled proteins. And just as long as these proteins that are being recycled all the time inside of them are normal proteins, then all the little peptides that are coming off of them would be peptides that our immune system has been trained to ignore or accept as being okay, right? So that's immune tolerance, or we should say self-tolerance of our immune system. So that idea of the cells having to present things in order for the immune system to see them is one way that cancer cells can stumble upon mechanisms for avoiding that. We discussed this idea that antigens, meaning any type of three-dimensional little molecular signal that can be recognized by the immune system, oftentimes those antigens are in the form of proteins that are expressed in different levels on the surface of a normal versus a cancerous cell. And we say that the expression levels of certain antigens that our immune system might be primed to look out for as evidence of a cancer cell, that has to do with the immunogenicity or the detectability of that cancer cell. In a kind of ironic way, cancers that are more immunogenic, right, means that they actually are more easily spotted by our immune system and trigger reactions of our immune system. Those highly immunogenic cancer cells are not going to be the ones that are very successful at being very aggressive, right, because they are letting the immune system see them and the immune system can attack them. So ironically, it's the cancer cells that are less immunogenic, that do not advertise their presence as much. Those are the ones that can sneak by the immune system. Some examples of mutations that allow cancer cells to evade the immune system include things like downregulation of expression of MHC proteins. So MHC proteins, you remember, are the little platforms on the surface of the cell that are actually carrying and presenting antigens like small peptides, right? So if a cancer cell stumbles upon mutations where it actually shuts down expression of these MHC proteins, then it's just not going to display on its outside evidence of it being cancer. Some mutations might involve upregulation of certain proteins that get secreted and modulate or inhibit the immune system from working. For example, the expression of TGF-beta that is received by lymphocytes, T cells that are coming to attack the cancer cell. If they get a big dose of this TGF-beta that's coming from the cancer cell, that deactivates the T cells and may actually cause those T cells to undergo apoptosis. Also, cancer cells might end up by accident learning how to express things like FASL. So FASL is the ligand for the death receptor, right? So again, cells around them then will start receiving doses of this FASL, and the cells around the cancer cells may start dying off. We also ended last lecture looking at overexpression of certain proteins on the surface of a cancer cell that may interfere with things like immune proteins, such as complement proteins, coming and being able to coat the surface of the cancer cell, causing the cell normally to rupture or just break apart due to complement proteins making all kinds of holes in the surface of the cells. So cancer cells may stumble upon the expression of things like CD59, which actually prevent the complement proteins from forming those Swiss cheese-like holes all over the cell. This last one we didn't get to last time, so we'll start off with it today. And it has to do with the recruitment of specialized T cells known as Tregs. Right. So Tregs are a special type of helper T cell. So remember, helper T cells are T cells whose job it is not to go around killing other cells, but to actually interact and communicate with other cells of our immune system to help orchestrate or coordinate a meaningful immune response. So some types of these helper T cells are regulatory, meaning when they bind to the surface of a target cell, they actually secrete inhibitory signals such that any other T cells that are coming and also recognizing that cell are inhibited from activating. So normally these Tregs, so these are totally normal T cells that are actually an important part of our immune system. The job of the Tregs then is to normally keep the immune system noise level suppressed. Otherwise, you'd always have T cells sort of accidentally activating all the time, and your immune system would always be in this kind of pseudo-activated state, right? So Tregs normally bind to targets and discourage other T cells that are interacting from properly being stimulated. So tumors then oftentimes stumble upon mutations that attract more of these Tregs to them. So that's what this cartoon is portraying here. So here we have the tumor cells, and the tumor cells have stumbled upon a mutation that they are now expressing the cytokine CCL22. CCL22 recruits Tregs. So these purple cells are the T regulatory cells, right? So they're receiving the CCL22 and they're attracted then to the tumor. And so they are now hanging out around the tumor. Right? Meanwhile, you have things like cytotoxic T cells or other helper T cells that have, let's say, T cell receptor on their surface that are trying to recognize certain types of tumor antigens. Right? So normally, these cytotoxic T cells would go ahead and kill these tumor cells, or these green helper cells would interact with the tumor cell and be alerted that there are tumor cells, and the green helper cell would then try to recruit other cells to go ahead and attack the tumor. But if you have a lot of these purple Tregs hanging around on the tumor, you can see here, they inhibit those other effector T cells from activating. At the same time, we mentioned that the tumor may have learned how to secrete high levels of TGF-beta. So TGF-beta not only helps to recruit more Tregs, as indicated here, but remember last time we discussed that TGF-beta will actually discourage the activation of effector T cells. So how important is this whole thing about Tregs in terms of in the clinic? It is very important because this is yet again another great example of a type of marker or type of pathology, that under the microscope that you can stain for the presence of certain things and use this as an indicator of a patient's prognosis. So here we have 3 cohorts of patients in this Kaplan-Meier plot who, when they first presented with their tumor in the clinic, the biopsy was looked at and stained for whether or not there were Treg cells hanging out around the tumor. And they broke the patients into 3 groups then. If they saw high levels or high concentration of Tregs already around the tumor, medium amounts of Tregs, or low concentration of Tregs. And you can see, right, those patients who had little evidence of these Tregs being associated with the tumor, their survival is much better compared to patients who have evidence of Tregs being recruited to their tumor, right? Either medium concentration or high concentration. Again, because the more Tregs that are already hanging out around the tumor attracted there because that particular tumor learned how to attract the Tregs, then those Tregs are stopping the patient's immune system from reacting against the tumor. Okay. One of my colleagues from USC, we were classmates in the PhD program there. He actually has a biotech company now in Los Angeles. And one of the things that they're trying to do is come up with different ways of preventing Treg cells from being attracted to tumors, right? So certain types of drugs that they're developing that actually involve antibodies that can interfere with the ability of Tregs to be recruited to the tumor. Right. So all of these things have to do with the tumor cell somehow finding a way to avoid being detected by the immune system, right? How it kind of slips by and either is under the radar so the immune system cannot see it, or comes up with clever ways of fighting back against the immune system and preventing the immune system from attacking it. Ultimately, though, if you can therapeutically boost the immune system's ability to better target and destroy the cancer cells, then that has really great advantages over just relying upon certain types of compounds, like small little molecules that are cytotoxic or kill cells, right? If you can leverage a patient's own immune system to do a better job, that's potentially the most powerful tool that you have to fight the cancer, because it uses or takes advantage of the fact that we have hundreds of millions of years of evolution backing up this system of our immune system being the second line of defense to attack cancer cells. Right? So remember, a patient's immune system may be perfectly fine, right? There may be nothing wrong with their immune system. It's the tumor cells that have come up with ways to sneak around the immune system. So if you can get the immune system to sort of wake up and pay attention to that tumor, right, then you might be in good shape to actually have a very positive and strong therapeutic effect. So one way to do this then is to utilize antibodies. So antibodies are these small little Y-shaped proteins that are made by the B cells of our immune system. They are part of our adaptive immune system, right? That means that the particular structures of antibodies are something that your immune system comes up with in the course of your life, right? It's not something that you were born with. And it's based upon all the types of things that you are exposed to during your life. And that's what we call it, our adaptive immune system. So clearly in the case of a patient who has a large cancerous tumor, that probably means that they don't have very good antibodies developed to detect those tumor cells, right? Like somehow their immune system has not actually focused on making the right antibodies that can label the cancer cell. So if we think about the wolf in sheep's clothing, right? That's essentially what we're talking about. The cancer cell on its exterior, for some reason, is ignored, right, and is allowed to keep doing its bad things. Right? So antibodies can be used therapeutically if they are directed against particular antigens that are on those cancer cells. And obviously that means you have to know something about the patient's particular cancer. So from biopsies and things like that, the oncologists can actually figure out what types of antigens are being expressed by this patient's particular tumor, right? And they can select for commercially available antibodies that are manufactured by pharmaceutical companies, right, as drugs. And so what those antibodies will do then, right, is they will essentially label or flag the cancer cell so that the patient's immune system now might pay attention to it. So monoclonal antibodies have become a very, very big multi-billion-dollar sort of area of drug development for pharmaceutical companies. I think a decade ago, there were not too many examples of monoclonal antibodies on the market, right? Certainly 20 years ago, you could probably count all the commercially available monoclonal antibody drugs, on your fingers, right? But now there are scores of them. And a lot of times, when you're watching TV and you see different types of drug commercials come on, these drugs are actually monoclonal antibodies that have been designed to target some sort of antigen presented by cancer cells. So they are monoclonal, meaning that they are generated oftentimes using an animal like a mouse, right? And then they are purified so that the antibodies that you have a high concentration of, right, are identical and they only recognize one particular signal. This is compared to polyclonal. So if you have a mouse, for example, and you injected a protein, let's say a protein that comes from a tumor cell that is unique on the surface of those tumor cells, right? That protein will have a three-dimensional shape that has several potential epitopes on it, right? Several potential little landscape features that the mouse's immune system could develop different antibodies to recognize. So as part of the way that the mouse's immune system would mount a defense against this, and which would be the same as the way our immune system would, right, is you would actually select different B cells that each make a particular immunoglobulin to bind these several different epitopes, right, on them. So in that situation, then your immune system would generate many, or I shouldn't say many, but a selection of different antibodies. Right? So you could say, well, that sounds good because then you have many antibodies that all detect the same thing. But the problem is the more different antibodies you have in a mix, if you're trying to use this as a drug, the more chance there is for any of those antibodies to have other unpredicted off-target effects. Right? So oftentimes when they generate antibodies for therapeutic purposes, right, they will isolate out the examples of the different antibodies that were made, right, test them each individually to find which one maybe has the strongest binding to its particular epitope, and then conduct all the necessary tests using that one antibody, right, because they can focus their research just on that one. So you go from a polyclonal situation down to a purified monoclonal antibody that only recognizes a particular target. Right. So as I said, the purpose of these monoclonal antibodies as cancer drugs then is to help focus the patient's own immune system to pay attention to whatever's being labeled inside of their body by this antibody. Right? And presumably the only thing if you've done all the necessary research and you've made sure there's no horrible off-target effects, the only thing that gets labeled in their body is the tumor cells. And that way, once the tumor cell is labeled, their immune system, which may be perfectly healthy, can then attack the tumor cell. Whereas before, their immune system, for whatever reason, was ignoring it. So I said that this is powerful because it makes use of the existing immune cells and pathways inside the patient, those mechanisms that have evolved for millions and millions of years, right, to be finely tuned to do such a good job of actually attacking things in our body that don't belong. So when you make these antibodies, then I mentioned they are oftentimes generated in animals like mice. So here we have an example of a typical antibody. So for those of you who took immunology, you remember an immunoglobulin like this is made up of 2 identical heavy chains and 2 identical light chains, and it forms this Y-shaped molecule. The ends of the arms are the variable regions. That's what the V stands for. These are the unique 3-dimensional shapes that can actually recognize a particular epitope or target. The stem of the antibody is the part that can then recruit some other cell and actually attack whatever cell this antibody is coding. So all the little Ys would be attached via the ends of their arms, coding the tumor cell. The stems would be sticking out, and those stems are then what other immune cells come and recognize and then attack the cell that's being covered. So in red here is the antibody that's generated, let's say, in a mouse, right? So its entire peptide sequence comes from the mouse genes that code for this antibody from the mouse's immune system. In contrast, in beige down here would be an antibody that comes from a human being, right? So the sequences are human sequences. So oftentimes, you don't want to just take a pure mouse antibody and go ahead and put it in high doses inside of a human. Why? Because much of this protein sequence that makes up the immunoglobulin, right, is different compared to the sequence that makes up a human one. So that means potentially the antibody itself could be recognized as a foreign target. And a person's immune system might just go ahead and destroy the mouse antibody. So what you need to do is play some tricks here, because you want to keep the parts of the mouse antibody that are the important parts, that are actually what the mouse's immune system came up with to recognize the cancer protein or cancer antigen that was injected into the mouse to generate the antibody. So you want to keep those parts, but the best scenario is if the rest of the antibody molecule is as close to being human as possible. So you can see here, one way you can do it is you can humanize this mouse antibody. What they've done here is they've looked at the parts of the variable binding domain that actually are the unique 3-dimensional shape that determine the antigen binding affinity. From immunology, you might remember that there are 3 loops of the peptide that actually determine the unique 3-dimensional shape. So what researchers can do is just take those 3 sets of sequences that make up those loops and go ahead and edit a human gene that codes for an immunoglobulin and put those in there instead. So that way you have a human antibody, but that has the correct 3-dimensional shape at the ends of the arms to detect the cancer target. Another way you can do it is just simply take the whole variable domain of the mouse off, right? If you cannot get away with just having small little peptide pieces moved into a human antibody, just take the whole sequence of the ends of the arms and put it on there, right? So we don't call that humanized anymore, right? Because that's what we call this, humanized, just taking little peptide sequences. We are calling this a chimeric protein, because now this is a protein that's a fusion of human and a mouse, right? So some of the strongest, best desirable monoclonal antibodies are the humanized ones, right? But a lot of times you can't get away with that, so you end up with chimeric ones. So this brings me to a discussion of the naming of these monoclonal antibodies. I'm sure all of you, when you watch commercials for a lot of these types of medications, it's kind of weird how they have very funny names, right? I mean, they always have a catchy kind of market name, like Rituxan or Gazyva or Arzerra, right? That's usually the big kind of name that you see in the commercial. But then underneath it, they usually have in parentheses or italics the specific, more sort of technical drug name. And it'll be things like rituximab or obinutuzumab or fatumumab, right? And you often think like, these names are so weird, right? Where do they come from? This is actually the proper nomenclature for it, right? See, these are not just crazy made up kind of weird names, but they actually mean something. So the first part of the name is, okay, something that the manufacturer makes up entirely, right? But the second part, the XI or the ZU or the U, have a particular meaning as well. The last part, the MAB, now you might realize that means monoclonal antibody, right? So any drug that you see that ends with a MAB, oh, this is an example of a monoclonal antibody that's used therapeutically. So this middle part, the Z, the ZU, the U, that has a meaning as well. That refers to whether these antibodies are chimeric, right? Whether they are humanized, or whether in reality they were antibodies isolated from a human to start with, right? And then they were able to then lift the sequence for making this antibody and clone the B cells from a person who actually make this antibody, right? So here are some other examples. I'm sure some of these in the course of your research, for your research paper, you came upon very common types of monoclonal antibodies that are being used as potent treatments in the clinic. So things like rituximab or trastuzumab, I think some of you have seen that one, or ipilimumab, right? So that's how the naming works. So let's take a look at the mechanism of how these monoclonal antibodies are doing their thing. Here's a good example where there are certain types of cancers that are lymphomas or leukemias involving the B cells becoming tumorous. Right? So B cells have on their surface certain types of proteins that can be used to identify this cell as being a lymphocyte, or even more specifically, a B cell. So one of these is CD20. So here is the surface of the B cell membrane. This dark protein here is this CD20 that's on the surface of B cells and other types of subtypes of lymphocytes. Okay. So the other cells in your body that are non-B cell, they wouldn't have this CD20. So therefore, if you want to go ahead and actually start attacking, right, all of the crazy, out of control, proliferating B cells that are part of this leukemia or whatever, the CD20 might be a good target to have an antibody against. So here are 3 of those antibodies from the previous slide: Rituxan, Arzerra, and Gazyva, or Gazyva. And so these, you can see here, this is the Rituxan, the reddish colored thing, right? And so that's the antibody. And I know you're looking at that and like, well, I don't get the Y shape, right? Well, maybe let's start with this blue one over here. It's easier to see, right? So the dark blue are the heavy chains. So you see the stem over here and then the 2 arms over here. And then the light blue are the light chains, right? So you can really see the Y shape here. And you can see the end of it is the right shape to pick up this particular 3-dimensional surface of the CD20 protein. Arzerra picks up a different 3D epitope or surface. And likewise, you can see how the Rituxan actually kind of picks up both. So here are 3 commercially available monoclonal antibodies, all 3 of which target the same antigen, the CD20, on the surfaces of B cells, but they target different epitopes of that antigen. So that's why you can actually have these drugs sort of competing in the marketplace. Okay. So once those antibodies bind to the surface of, let's say, a malignant B cell, so here's just as an example, using the Rituxan or the rituximab, right? Here's the antibody. It will coat the surface of the malignant B cell, and then it might employ two different ways of attacking this B cell. One is, as I said before, the stems of the antibody. So imagine now, here's the CD20 protein on the surface, and now let's imagine that the CD20s are now coated with these dark brown rituximab antibodies, right? The stems of those antibodies can then attract some other type of immune cell, right? So this method, this type of attack is often called ADCC, which is a antigen-dependent cell cytotoxicity, right? Because then a macrophage or some other natural killer cell that normally might have floated by and ignored this cancer cell, because obviously that's what's been happening for years in this patient's body, is for some reason their cells are not paying attention. Now suddenly they take an interest because they see the antibodies, right? So that's one way that the tumor cell gets killed. Another way is that the coating of it with antibodies attracts complement proteins, right? And those complement proteins can then go ahead and build these little holes, these membrane attack complexes. And so then the whole surface of the B cell becomes covered with little holes and the cell lyses. Technically, this shouldn't say apoptosis, because if you're just rupturing the cell, that's not really apoptosis, right? It's the cell basically blows up because all the water rushes in and all of its guts kind of spill out. Right? This upper one up here would be more properly termed apoptosis, because that's what the macrophage, oh, not the macrophage would just start eating away at the cell, but a natural killer cell or a T cell would go ahead and kill this cell. Right. So I mentioned that because different monoclonal antibodies can target the same antigen protein, but actually be designed to target different epitopes on them, you can use them in combination then, right? Because that seems pretty logical, that you don't have to just have one antibody being used as a treatment at the same time, right? If you have other ones that also target the same thing, but different epitopes of it, then you can combine these drugs. So here again, here's another example. One tumor-associated antigen is the EGF receptor. Remember, all the way back early in the semester, we were learning about receptor tyrosine kinases on the surfaces of cells. So we learned that these receptor tyrosine kinases help take in mitogenic or stimulatory signals that encourage the cell to grow. So normally a healthy cell should have a limited amount or limited number of these EGF receptors on their surface. Oftentimes, cancer cells will overexpress or make too much of these EGFs. And we even learned before that if you have way too many of these EGFs, then they might start dimerizing on their own and turning on, right? So we can use EGF as a target with monoclonal antibodies. So one EGFR molecule, right, one receptor molecule, has many potential target surfaces or epitopes. Let's look down here. So here's a HER1 or EGFR family member receptor. So here's the cell membrane, right? And it has all this sort of complicated three-dimensional shape. This particular antibody over here, cetuximab, you can see, I think they're only showing the arms of the antibody. They're not bothering showing the stem. But the idea is the two parts here are the heavy chains that are making the arms, and then the yellow ones are the light chains that then together with the heavy chains make the arms, right? You can see in this case, the cetuximab binds to this epitope over here, or this unique three-dimensional surface. We can just call it epitope 1. If we look at other examples of EGFR, so this is HER2/neu, which we studied before, right? So trastuzumab. So I know some of you were doing research on breast cancer, so you probably came upon trastuzumab as an example of a very popular therapeutic drug. The market name is called Herceptin, right? So now you kind of can figure out it's very clever because it's a HER2/neu. It's based upon the HER2/neu receptor. Right? So trastuzumab, you can see here, it binds to this epitope over here, right? And this other one, this pertuzumab, binds to this epitope up here. So you can use these monoclonal antibodies in combination then, right, to really try to target different parts of the antigen signal on the surface of the cancer cell. Another way that monoclonal antibodies can be used is not necessarily by labeling a tumor cell so that the immune system recognizes it better. One thing that I've already kind of hinted at, and we kind of talked about, is antibodies can actually be used to interfere with other proteins being able to, let's say, properly interact with a receptor. So here's an example of receptors that are involved in T cells activating, right? So that a T cell might turn on and kill whatever cell that it's pressed up against. So T cells recognize their targets using their T cell receptor. So here on the cartoon on the left here, let's say this cell on the top is a naive T cell, right? It's not activated yet. It has on its surface its T cell receptor, and that T cell receptor is the right shape to detect a particular type of antigen. A naive T cell, in order for it to be activated, it's not enough that its T cell receptor sees something that it recognizes. It needs to have some additional encouragement to activate. And that additional encouragement comes in the form of the cell on the bottom, whatever cell's presenting this antigen, also having certain signals that the T cell can use as a convincing signal to properly activate. So in immunology, we study that antigen-presenting cells, like dendritic cells, have this B7 protein on their surface, and that B7 is received by the T cell on its surface by the T cell's CD28, the light green, right? So not only the T cell receptor in blue seeing the antigen that it recognizes, but at the same time, the CD28 receptor seeing B7, that combination of two signals, so the red arrow and the blue arrow, combine and activate the T cell. Right? Likewise, that T cell also has a certain number of inhibitory receptors on its surface that also bind to B7, but do the opposite effect. So one of those is CTLA4 in red here, or in orange. Right? And so if the CTLA4 binds to the B7, it actually sends inhibitory signals to the cell. So what we're talking about then is the relative balance of how many CD28 receptors in light green versus how many CTLA4 receptors in this orange color are trying to tell the T cell sort of conflicting messages, right? But whichever one wins out is what the T cell will do. It will either activate if it sees more from the CD28, the light green receptor, or it will stay shut off if it sees more signal from the CTLA4. So this is where our discussion of antibodies come in, because if that very important interaction is between two proteins, in this case, the B7 of the presenting cell and the CD28, or the B7 and the CTLA4, an antibody that is designed to actually recognize that interaction surface might then bind to one of these or the other and interfere with proper interaction. So let's see some examples now of how might antibodies designed against these molecules affect the T cell in terms of its activation. So here is a commercially available monoclonal antibody called Yervoy, funny name, right? So this is ipilimumab, right? So ipilimumab is actually an anti-CTLA4 antibody. Why? Let's just move back one slide again. Because remember, CTLA4, it's this receptor. If it is engaged by B7 of the target cell, this sends inhibitory signals so that the T cell does not turn on. If you can interfere then with the CTLA4 sending those inhibitory signals, you have a better chance of T cells activating. Right? So you can imagine little antibodies that kind of stick right in here against the CTLA4 surface, and here might get in the way of the CTLA4 properly being stimulated by the B7. And that's what this ipilimumab does. So here is an early mouse experiment showing the effect of this anti-CTLA4 antibody. What they've done is in these mice, they've gone ahead and injected tumor cells into the mice. So these are lab mice where they're injecting some tumor that they know the tumor cells will grow. And then following the injection, they go ahead and give the mice antibody treatments, maybe 3 different sort of doses or 3 different separate occasions where they put this antibody in. And they're doing 3 different sort of experimental groups here. One group is the no treatment group. So this is when you inject maybe just like some saline or something like that as a vehicle for control. And you follow then the growth of the tumor over, let's say, a month or so, right? Because this is days after injection. So if you follow the red line, this pretty much tells you, okay, normally if you're not interfering with these tumor cells and you're not interfering with the mouse's immune system, then this is the expected growth of these tumor cells inside of the mouse, right, with no treatment. If you give these mice anti-CD28 antibody, all right, so again, let's back up real quick. Remember, CD28 is the light green receptor here, right, that normally needs to be properly activated so that these red arrows happen, and those red arrows work with the blue arrow to turn on the T cell, right? So the T cell might now go ahead and do something to be alerted to this being a cancer cell. If you interfere with the CD28, right, you might imagine that you will actually hinder or inhibit the numbers of T cells that properly activate and see the tumor cell. And in fact, if we move back to the experiment here, that's what you see, right? So the blue antibody messes up the ability of T cells to properly activate. So you can see as a result, the tumors actually grow much faster, right? Because the existing T cells are not activating at the level they should be, right? But really the therapeutic part of this, right, that blue line is more of like a really neat sort of comparison, because really the thing that matters is the green line. And that green line is if you give these mice the anti-CTLA4 antibody, right? So now you're preventing the CTLA4 receptor from activating. And so therefore, fewer of the T cells get the inhibitory signal. And likewise, more of those cells then will properly activate. And as a result, this is a pretty stunning result, right? Because by preventing the CTLA4 receptor from being engaged, because you're now gumming it up by having this antibody, you can see what a dramatic impact that has on the tumors, on limiting the tumor growth, right? So here is now on the right, some real human data. So this is now in the abdomen or the thoracic region, chest region of a patient. You have this lung tumor right here. Okay. Oh no, sorry, this is actually an organ. This is the tumor mass over here, right? The main tumor mass, and you can see it over here as well. Okay. And this is pre-treatment, before they started on this ipilimumab, and this is 5 months after being treated with the ipilimumab, and you can see dramatic reduction in the abdominal tumor size. So there are other immune response-based cancer therapies that aren't necessarily involving antibodies themselves. So one approach is to transplant bone marrow. In other words, if you want to go ahead and get rid of the problem, if the problem is actually a cancer of B cells or something related to the immune system, maybe you want to just eliminate that patient's defective immune system and reboot it with a healthy immune system, right? So you can do that. If you give the patient a high dose of radiation to kill off the hematopoietic stem cells that are generating these problematic cancerous lymphocytes, you can then replace it, hopefully, right, with healthy stem cells. Number 2 is they found that certain types of bacteria can, for some reason, wake up the immune system so that the immune system becomes more alert and then might start realizing or seeing things like tumor cells that previously the immune system was ignoring. But the mechanism behind this is kind of poorly understood. And as you can imagine, the idea of giving somebody intentionally a bacterial infection in order to hopefully get their immune system to see their cancer maybe isn't the best solution, right? The third one is actually vaccinating against cancer. So this is a very new thing, this idea of actually coming up with vaccines against cancer. But here are two examples of them. Gardasil, which is now a vaccine given kids as they're approaching their tween, and teen years. Gardasil protects against HPV infection. And Provenge is a drug that's being used on older, middle to older age men to fight against prostate cancer. So in the remaining time, let's quickly take a look at how these two types of vaccines work. So cervical cancer is linked, without a doubt, to human papillomavirus. Right? This is one of those great examples of cancers that have an indisputable link to viral infection. So HPV is the most common cause of sexually transmitted diseases. About 50% of sexually active individuals actually harbor HPV. The majority of time, acute HPV infections kind of clear away and you become asymptomatic after some time, right? But there is residual virus genome that has integrated into cells of the body. And so in this case, so here we have the HPV particle here. Here we have cells that have been hijacked by the HPV virus. So they are producing more HPV virus particles. As we've discussed previously, these are not the cells that you worry about with cancer, right? Because these are the cells that are actually just blowing up and releasing virus particle. Once the infection is cleared, the cells that you worry about are the ones that got like random integration of the HPV genome or fragments of it in such a way that they never produced more virus particle, but they are now on that path to transformation. Right? So if you have a vaccine that protects against HPV infection, then you're really targeting the main cause of cervical cancers. So Gardasil is a vaccine that's made with different peptides that represent this L1 protein. That's one of several different proteins that are on the surface of the virus particle. So Gardasil is actually made of 9 different possible antigens coming from different strains of HPV 16 and 18. Right, those are the strains that are most likely to cause cervical cancer. And so Gardasil 9 has been shown to have 100% effectiveness in preventing precancerous lesions of cervical tissue. So it really is our first example of a vaccine that can be considered a vaccine against cancer, right? Because it's so effective in protecting against the main cause of that cancer, the HPV virus. But Gardasil is controversial, right? Because the only time to receive a vaccine in order for it to be effective is before you've ever been exposed to the virus that the vaccine is meant to protect against, right? And so that means you want to vaccinate kids before they become sexually active, like in their teenage years. And so that's why I think a lot of families and parents don't want to really consider it, because they don't want to think about their 9 or 10-year-old kid or 11-year-old kid as being sort of like, oh, one of those kids, right, who are sexually active, right? And they sort of miss the point of how vaccines work. The last example then is this Provenge that attacks prostate cancer. So prostate cancer cells, as shown here, secrete different types of tumor-specific antigens. So these are things that you'd only find in prostate cancer cells. One of these is this prostatic acid phosphatase, or PAP, right? That's what's stained in red here. So what researchers have found is that if you want a person's immune system to really pay attention and start attacking their prostate cancer cells, because for some reason the immune system is ignoring it up until now, this PAP antigen is a great target, right? And so what they do is they take the PAP, so this is red diamond, and they fuse it with another protein, in this case, CSF, or colony stimulating factor, which is the signal that's taken up by monocytes to help differentiate those monocytes and turn them into things like dendritic cells in the immune system. So on the left here, you see a monocyte in yellow. It doesn't care about the red diamond part, the PAP, right? Because that's not part of what a monocyte normally would pay attention to, but it loves seeing the CSF, the blue part, right? Because that's what the monocyte is designed to recognize by its CSF receptors. And this then stimulates the monocyte to turn into a dendritic cell. The dendritic cell, again, is the cell that's responsible for presenting all kinds of antigens to other immune cells to get them to wake up and pay attention. So in this case, the drug, the fused PAP CSF, gets taken in by the dendritic cell. The dendritic cell processes the protein, turns it into small little peptides, and loads up those peptides on MHC class 2. And MHC class 2 then goes ahead and presents the small little peptide, this PAP, that comes from this PAP protein, on the dendritic cell surface. So how does this work? Basically what they do is they take a prostate cancer patient, they take lots of their blood out and extract from their blood their own monocytes. Then in the lab, they do this, feeding the monocytes this fusion PAP CSF drug, and it trains their monocytes in vitro, basically in a test tube, to be really, really good at presenting the red dot, right, the PAP antigen. Then what they do is, after they've had these super well-trained dendritic cells, they put them back into the patient's body. And then these dendritic cells go into the lymph nodes and they start really showing the immune system, hey, forget what you guys were doing before. This little red dot, this PAP, is the thing that we need to pay attention to. And then the person's own immune system, then the T cells go to work, right? And they start attacking the tumor. So, this is a great example of leveraging the patient's own immune system to fight their cancer. The drawback of this is, if you were sort of paying attention to the process here, is that it is tremendously expensive, this type of treatment, right? So I believe the last figures I saw was that a typical 3-dosage treatment of using Provenge. So Provenge is not necessarily the little drug itself, it's actually this whole procedure, right, is patented as a therapy, Provenge, which I love the name because it's revenge against your prostate cancer. But each treatment is like $90,000. So in order for a patient to undergo this, it's close to like $300,000, right? And unfortunately, at this point, it's not covered by insurance, right? Because something like this is still considered very experimental. You can imagine, though, in the coming years, these types of treatments will become more commonplace and less expensive, right? And these do have big advantages over just relying upon certain types of little drugs that simply attack cells directly, all right? So hopefully that gets you kind of excited thinking about great potential for directions that drug therapies can go in the next few years that you might end up being involved with.